The second type of chromosomal rearrangement we'll consider is chromosomal deletion. This is not the deletion of an entire chromosome, but rather just a part of it. If we take our homologous chromosomes and delete regions 4 and 5 on one of them, the resulting cell is heterozygous for the deletion, having only one copy of genes in regions 4 and 5. There are several ways that this can happen. Let's examine one of them. As we saw on the last page, unequal crossing over can cause a duplication. Unequal crossing over can also cause a deletion. This time we won't involve the centromere because if we did, we'd just lose one of the chromosomes. Again, deletion is the loss of just part of a chromosome, not all of it. Let's say our two homologous chromosomes misalign, like this, which again occurs because repetitive sequences at these points can align in multiple ways. If unequal crossing over occurs, one chromosome will be 1, 2, 3, 3, centromere, 4, 5, 6. And the other chromosome will be 1, 2, centromere, 4, 5, 6. So we see that region 3 is duplicated, while in the other chromosome, region 3 has been deleted. If this chromosome is inherited by the offspring, and the other chromosome of this type is normal, the offspring will only have one copy of genes in region 3, the genes in region 3 that are coming from the normal chromosome. So, of course, deletion alters gene dosage. If a cell is heterozygous for a deletion of regions 5 and 6, the cell only has one copy of all the genes in these regions, including our hypothetical A gene. So the gene dosage for these genes is only 0.5x. One copy will only produce half the amount of protein. Whether this produces a mutant phenotype or not will depend on what gene or genes are deleted. Recall that the term haplosufficient means that one copy of a gene is enough to carry out its function and display a normal phenotype. So if a cell has one of its two copies deleted, as we have here, and that gene is haplosufficient, no problem. No mutant phenotype occurs because one copy of the gene is enough to carry out its function. In other words, being haploid for that gene is sufficient. But if a gene is haploinsufficient, then we've got problems, because there isn't enough protein to fulfill that gene's function, and we might see a mutant phenotype. Many human disorders are caused by the loss of one copy of a haploinsufficient gene. Here, try to answer the question, what would the consequence be of a homozygous deletion of a haplosufficient gene? I'll give you a moment to pause the video. Okay. Whether a gene is haplosufficient or haploinsufficient, it's fairly likely that we'd see some kind of mutant phenotype if an organism was homozygous for a deletion, because it would then have 0 copies of the deleted genes, and therefore make none of that protein. How harmful the mutant phenotype will be, of course, depends on what gene or genes is deleted. Now, as we said on the last page, if the centromere is deleted, the spindle fibers will not attach to the chromatids of that chromosome, and during anaphase, they won't get pulled to the poles of the cell. And they might not get enclosed by the new nuclei and get destroyed. This would lead to monosomy, which is actually an aneuploidy. We'll have more to say about aneuploidy in a later video. The point here is that deletion of the centromere usually eventually leads to the loss of the whole chromosome. Deletion can also lead to something called pseudodominance. Here are our two chromosomes. Let's say our A gene is in region 5, and that this cell is heterozygous, with this chromosome having a recessive small a allele and this one having a dominant big A allele. Initially, this cell will have whatever phenotype big A confers because it's dominant. But if regions 5 and 6 are deleted from the bottom chromosome, but not the top chromosome, the big A allele is lost, and the small a allele is the only allele of this gene left. Since it is no longer being masked by the dominant big A, the small a confers or expresses its phenotype, even though it's recessive. This expression of a recessive allele after the deletion of a dominant allele is called pseudodominance, and in this way can actually change the phenotype of an organism. So to sum up, deletion can alter gene dosage, lead to aneuploidy, and the expression of recessive alleles. So that's it for deletions. See you all in the next video, which covers inversions.